Hello and welcome. You know, about 15 billion years ago, the whole universe was confined at a single point. And then an explosion happened, and we call that explosion Big Bang. So, what happened that everything, mass, space, time, everything, energy, was confined at a single point. The whole universe was at a single point, and then an explosion happened, and because of that explosion everything that was there inside that small condensed uh, form of matter, energy, space and time, everything was there. And because of that explosion everything started going apart from each other. In the same way, when you witness a firecracker, when it blasts, you think all the component of firecracker go away from each other. So if you think about the whole universe, all the stars, all the planets moving around those stars, so everything is going away from each other. And if you can see that things are going away from each other with time, and if you if you draw a fine line, extrapolate it to the past, it seems that whole universe was confined at a single point. And explosion happened, and everything started going away from each other. And somehow universe came to this point that we witness nowadays, that sky is full of stars, and and planets are revolving around the stars. Sun is a star, and around the sun about 8-9 planets are revolving, and around the planets multiple satellites are revolving. And this is how things happened as per laws of physics. One day I was talking to one of my friend, and I told her that I want to go in the sky on and be at some star, and then have fun out there. Sky is so beautiful. And she answered that when you will go to the sky, then on some star, then you would not see the sky in the same way you see it from here. Because when you will go there on some single star or some another planet, then you will see this place, including our earth, moon and all these places, look like the same that you witness from earth to that point. You got my point, that we do not need to go to some other planet to have fun. We are already in this galaxy, in this universe, and witnessing whole universe. Even if you go on some other planet, you will witness the same thing far away from that place, and there you will feel the same, even worse than this, because here on our earth we have water, we have plants, we have animals, we have human and a lot of things. But there is no guarantee till date that you will find similar kind of environment out there. But the reason most of the people like to go to the sky on some other planet is just because we think that how beautiful the sky is. But sky looks beautiful only when we look at the sky from a distance. If you go there, you'll again witness other part of sky, other part of universe, and that part will look beautiful. So presently wherever I am, this universe, this part of universe or the part of sky is indeed beautiful. It's just that we do not see where we are. We see, that distant sky and think that that sky is beautiful. No, we are in the same universe, and we do not need to go anywhere. Earth is beautiful, and from here witnessing rest of the sky is beautiful. But today's topic is not about astronomy. Today I just wanted to introduce something we keep on calling cell. What is cell? So if you think cell is a simple thing, then this video is for you. And if you think cells are very complicated thing, then this video is indeed for you. Let me put it in a way that why I am saying this. So what happened about 15 billion years ago, Big Bang happened, and I'll be looking at the monitor, computer monitor, and probably you may think that I'm not looking at you, but I'm doing some work also. So I'll look at there and I'll keep on talking with you. So about 15 billion years ago, the Big Bang happened, and because of that Big Bang, everything, matter, space, time, started from that point. And you imagine when whole universe, all the matter, all the dark matter, dark energy, space, everything was confined at a single point, then how dense it would have been. So we assume that it must be very dense. And then what happened? Multiple massive structure formed, and Earth was one of them. And so this complete solar system, our solar system has sun and 8-9 planets revolving around the sun. So the point is that when this solar system formed and all other solar system formed, so all the planets, all the stars are still highly energetic flammable substance. So if you study sun, it's nothing but a lump of a big collection of helium and some other traces elements, and helium is going through nuclear fusion, and because of nuclear fusion there is a huge amount of energy is getting released from the sun, and that's what make it a sun. So a source of energy. Sun is just a source of energy. And similarly at that time Earth was also was in heat bath everywhere. It was so damn hot, like it was under fire. And what happened that at that time when Earth formed after the Big Bang, so it was just matter in fire, and somehow there was cloud formation that happened, and then it kept on raining for million of years. And what happened when there was rain? So rainwater first of all cooled down the earth, and second of all that wherever there was some earth has a irregular shape, even it is spherical or whatever the shape of earth is, bit flattened on the top and then a spherical thing. So there was water got collected in an area where there was where they can find lower free energy, what I mean. And then ocean formed. Okay, and when ocean formed, earlier it was hot, and with time it started cooling down. Okay, and in that ocean life originated. So about 3.6 billion years ago life originated. So how did that happen? So we had carbon and oxygen, and due to reaction between carbon and oxygen, CO2 formed, carbon, hydrogen reacted with each other and formed methane, and then there was formation of ammonia. So these things formed first in the ocean, ammonia and methane, these things. So and then what happened, these ammonia and these methane and some carboxylic group, they interacted with each other, formed some covalent bond with each other and formed some amino acids. And then also there was formation of RNA, and RNA remained on this ocean, ocean on this planet. I'm talking about the Earth. So RNA remained here for a long period of time, and that part we call RNA world. And then there was formation of DNA, and then and these things took million of years time. I'm not going to give you the time scale when they developed, but yeah, they formed at that time, and these smaller amino acids then joined with each other and formed some protein, and then DNA formed, and then RNA formed at that time. RNA was earlier than protein world and the DNA world, because RNA has a capacity to, RNA can act as genetic material as well as the enzyme. So that's why RNA, there is an evidence. This is indeed an evidence that RNA evolved before than protein and DNA. But going ahead, so these DNA, RNA, protein, they started meeting with each other and they interacted and they started forming some assemblies, and in the beginning we call them conservates, like some smaller structure formed, and when they met with each other. So the thing is, whenever some interaction happens anywhere, even in living system or in the non-living system, everywhere, because of interaction, some emergent properties come out. Like if oxygen has some property, hydrogen has some different property, but when they meet in some fixed ratio, then they form water, and water has a property which is completely different than hydrogen and oxygen. So this is called emergent properties. In the same way, carbon had different property, hydrogen had different property, and when they met, they formed methane, and methane had completely different property. In the same way, ammonia and carboxylic group and this carbon, hydrogen, they met and formed amino acid, which has different, totally a different property. And when RNA formed, and then DNA formed, and then these protein formed, and these things interacted, and whenever they interact, they make some super assembly, and that super assembly has some emergent property which is different than its constituent elements property, constituent partners property. And because of this emergent property, the conservates had totally different property than independent RNA or DNA or protein. And then these things started combining with each other. There was separate organism. Some bacteria formed, which is some in the initial phase of evolution. And bacteria are prokaryotes. And simply prokaryotes are also assembly of DNA, RNA, and protein, but they didn't form well defined boundary to perform different function. However, if you take out my cell and look into the microscope here, you will see there are different boundary inside the cell where different functions are getting performed. If you have to do, if cell has to do DNA replication or DNA to RNA formation, that we call transcription, then there is a fixed bonded environment which is called nucleus. So inside the nucleus you have these reactions happening. If you have to join amino acid one by one and form some protein, then there is an environment at ribosome. However, ribosome is a membraneless organelle. if you have to perform some respiration reaction, then there is a separate environment surrounded by the boundary is in cells make their boundary using phospholipid and some protein. We make these boundary, like if you have to build a house, then you use bricks and cement to make this boundary, but cells use phospholipid and protein to make such boundaries. So if it is a mitochondria, if cells have to do respiration reaction, then they have a separate environment we call mitochondria. Cells, if they have to keep some acidic bag inside them for various purpose that we'll talk later, then they have a separate environment for that surrounded by a boundary of phospholipid and protein that we call lysosome. If they have to traffic something out of the cell, then they have different kind of structure that we call Golgi body. So if you look at my cells, which is an advanced form of cells, as I am a eukaryotic organism, then there are multiple bonded environment completely dedicated for different kind of function. But when bacteria evolved, these prokaryotes evolved, they do not have these bonded environment to perform different kind of activity. So they have some nucleoid like structure which is in association with cytoplasm. They have ribosome doing some function in the cytoplasm. They perform their respiration in the cytoplasm only. They have genetic material sort of exposed, or maybe a vivid structure there. We call it nucleoid in the bacteria. And since these organisms, bacteria and prokaryotes, were primitive organisms, they still do not have well defined membranes. But evolution happened, and unicellular eukaryotic cells formed. And I'll define what do I mean by a cell. And these eukaryotic unicellular cell then again interacted with another cell, and let's say that some unicellular cells engulfed some bacterial cells, and that bacteria was some aerobic bacteria. Aerobic bacteria means bacteria which has capacity to do some respiration reaction using oxygen. So when I say capacity, I essentially mean that they have the machinery, the protein machinery to do that function. So if you think about respiration reaction, then to perform a respiration reaction, you require multiple protein, you require multiple enzyme, and those bacteria had these kind of enzymes, these kind of proteins, hence they could independently do respiration reaction. So what happened? These cells engulfed those bacteria, and the bacteria came inside the unicellular cells. So it had a symbiotic relationship. Like those aerobic bacteria got some protection and shelter in the cells, and cells will get energy, ATP, in response to that. So this is how, and there was some bacterial cells which has capacity to do photosynthesis, because they have enzyme and protein machineries to do photosynthesis reactions. Now what happened that when these bacteria, photosynthetic bacteria cells was engulfed by some eukaryotic or to be eukaryotic cells, then they started acting like a chloroplast. So bacteria in a eukaryotic cells, we call them chloroplast if it is plant cells, or some algae which has chloroplast. And mitochondria was, during the evolution, it was also sort of bacterial cells, but when it is inside, well developed cells, then they started doing some function. That function is respiration reaction, and we call them the mitochondria. So this is a kind of collaboration we also do in our life. We collaborate with each other to when we collaborate, we essentially want some symbiosis, like if the collaboration should benefit both the partner, and that's what happened. Okay, so now evolution happened, like these cells were not made in the air by some god, but these cells are the result of a long time during the course of evolution. But today, and then one cell formed, and then cells aggregated with each other, they associated, they did collaboration with each other, and then they started forming some higher organism. So there were three branches from single cell. One was for plants, so all lower plants and higher plants. If you think about bryophytes, pteridophytes, thallophyta, bryophyta, pteridophyta, angiosperm, gymnosperm. So there is one branch for the evolution of plant, and another branch is for the evolution of animal. So we have, if you think from the beginning, we have protozoans, and then porifera, then coelentrates, then this annelids, arthropod, mollusk, and soon reptiles, and then developed till mammals, that we are the vertebrate and invertebrate. So vertebrate also evolved, and third branch was for the fungus. Fungus also developed. Some fungus were having septa, some do not have some septa, and there are different kinds of functions. And these three branches evolved from here, which is the protistans. Protistans were cells which has unicellular. So these are protists are unicellular eukaryotic organisms, and then it is somewhere linked with prokaryotes, means bacteria. And prokaryotic cells, as I said, that it's a primitive cells which were evolved long back during the evolution, in the early time of evolution, and they lacked separate environment to perform different kind of chemical reaction which is needed for survival of a cell. And then from prokaryotes there are protista, eukaryotic unicellular organism, and then three branch developed. One is for plant, one is for animal, and one is for fungus. Now if you look at any cell, any eukaryotic cells, then what do they have? First of all, they have a plasma membrane. So if we talk about cell, even bacterial cells have some wall which is made up of phospholipid. Of course, in order to segregate from rest of the universe and call yourself a cell or something, you have to make a boundary, right? If let's say if I die and my whole body disintegrate into the environment, then where am I? And there is another philosophical definition, that there is a still I that exists even without the body. But that is the true consciousness. I'm not talking about that. I'm talking about the basic cellular machinery that our body has. So in order to call my body a body, I need to have a skin to separate myself from the rest of the environment. If my kidney and liver and heart are disposed in this whole city, then I cannot call I am this. No. So that's why cells have a separate boundary made up of phospholipid and proteins which separate a cell from rest of the environment. And of course a cell require a separation from rest of the environment to perform live reactions, and these live reactions are called metabolism. So inside the cells, thousands of I will say that millions of reactions happening all the time inside the cell. and these reactions require a specific environment. You cannot perform these reactions everywhere. So when I say specific environment, what do I mean by that? So to perform different reaction, you require certain pH, fixed pH. Like if you think about protein synthesis happening, then it requires a 7.4, 7.2 pH. If you want to do the same reaction in a pH, acidic pH of 1.5, then you won't be able to perform that reaction. So although metabolic reactions should be called as live reaction or not, it is a separate debate, because the metabolic reactions that happen inside the cell, same reactions can be performed in test tube in the lab. Then should we call that we have performed some live reaction inside the test tube? I don't think so. So these are simply reactions, multiple reactions happening inside the cytoplasm as per the laws of thermodynamics. And since these reactions, in order to for this reaction to happen, you require a specific environment, and that's why there is a boundary which just to maintain certain kind of environment inside the cell, and these boundaries are made up of phospholipid. So if you look at the structure of cell, so what happens, there are phospholipid. When I say phospholipid, so there is a phosphate group, and phosphate group is is a polar compound. There is a phosphate group, and phosphate group is a polar compound because it has charge, and charge makes it polar, because water is also polar, and since cells evolved in a polar environment, which is water, so they must have, if this is a cell, let's say if this thing is a cell, then the boundary, upper surface of it must be polar, so that it can remain in a polar environment of water. Otherwise let's consider opposite thing. Like let's consider all the upper surface of cell is hydrophobic non-polar. Then what will happen? All hydrophobic non-polar thing would like to go away from water and make a clump away from the water. So in order to and in the same, when cells evolved in the ocean, so they today also, if you look at RBC cells, so they are floating in your blood, and blood is indeed 70% water, and which is a polar environment. So since cells evolved in a polar environment, the upper surface of it must be polar. So the nature and so there is two design always, and I'll keep on comparing. So if I talk about a microscope, if I talk about a cell phone, and if I talk about a computer, and if I talk about the electricity system in this room, it is completely engineer's design. And when I talk about cell, it is the nature's design. So nature designed in a way, and when I say nature's design, I essentially mean evolution. Okay, so the nature design is such that it has to be, since cells evolved in water, it has to be the upper outer surface of cell must be polar, and if it is polar, then what is the suitable partner to make this polar upper surface of the plasma membrane? That is phosphate group. Now second thing, that when I talk about cell, and I call, let's say this is the cell, then upper environment is water, even if it is in ocean, and even RBC floating in your blood, or even if there is a skin cell, still there are water. Take it as a rule of thumb that water is present around every cell. So that is why the outer surface of cell has to be polar, and phosphate is a negatively charged group, which makes it an excellent polar group. As well as inside the cell, the cytoplasm is also 70% water, and the cytoplasm is 70% water. That makes it polar, and hence the inner surface of this wall, that is the plasma membrane, has to be polar again. So if this is the cell's outer wall, which is polar, made up of phospholipid, then cell's inner wall, let's say this thing, this is the inner wall of cell, this also has to be polar, because cytosolic environment is also very polar, and hence the inner surface of cell is also made up of phosphate group only. So this is one thing. Now the question is, if outer surface and inner surface is polar, then water will interact with this polar group and these things will be dissociated. In the same way, if you put some polar molecule, like if you put NaCl in water, what happens? They get dissociate from each other, because NaCl exists as NaCl, sodium chloride, because of electrostatic interaction force that exists between Na plus and Cl minus, and the value of this force is, if you understand electrostatic force, is 1 by 4 pi epsilon naught epsilon r q1 q2 by r square, and this q1 and q2 is charged on sodium ion and chloride ion respectively. R is the distance between them. 4 is a constant, pi is also a constant, epsilon naught is permittivity in the vacuum, and epsilon r is the permittivity in water, or in the medium, wherever the medium is. So if it is air, then relative permittivity is 1, and if the medium is water, then relative permittivity is 81. So NaCl in air, when it is kept in a dabba, then it has force which is 1 by 4 pi epsilon naught q1 q2 by r square, because epsilon r is 1 here. And if you put the same NaCl in the water, the value of epsilon r becomes 81, and hence the force of attraction between Na plus and Cl minus reduced by 81 times, which dissociate Na plus and Cl minus away from each other, and hence they basically force decreases, so they will dissociate. Force of attraction will decrease, and then water molecules come and they surround Na plus and they surround Cl minus separately, and then they stabilized by the polar water molecules only, and they are no longer doing this work. They are no longer together. In the same way, let's assume that if nature would have designed the cell membrane such that there is a phosphate group at the top of this surface, and there is a phosphate group at the bottom of surface, then it would have been dissociated by water. So there was a need to put a hydrophobic glue between all these two phosphate groups, and nature designed in a way that they took lipid. Okay, so what did they do? They just made it like this. They just made the boundary like this. So if I can demonstrate you just for the visual understanding, so like this. So this is your phosphate group here, and this is your phosphate group here. So if this is a cell boundary, and there will be multiple, so this is the phosphate group in the association with cytoplasm, and this is the phosphate group in the association with the water which is present outside the cell, and these two thing that you see is made up of lipid. So that's why we call the phospholipid, and that's why this is another phospholipid. And these lipid tails are towards each other, because they form a very nice hydrophobic layer, and in order to stabilize it in a water environment, water is outside the cell, water is inside the cell in the cytoplasm, there has to be a polar group. So phosphate group is here as well as phosphate group is here. and in between there are layer of hydrophobic layer of lipid, and we call this thing phospholipid, these two molecules of phospholipid. But remember, here I am showing you only one tail, but in reality there are two tail of phospholipid, there are two tail of lipid and one phosphate group, and hence so these two tails has some importance that I'll not talk today, but maybe some day later. Okay, so what I said that when cells evolved, nature separated the cell environment from outside environment by something called phospholipid, where phosphate is outside the cell in association with water. Whether it is in, during the evolution it was involved in ocean, and that's why today also around every cells we have water. We are 70% water only. Now this is one thing, and to make an association with cytoplasm we have another phosphate group, and in the middle this thing is made up of lipid. Okay, it has two tail, it is making like this. Okay, and this boundary, this layer of plasma membrane or biomembrane, whatever you want to call it, separated the cytoplasmic environment from the outside environment. Now before I go into the cytoplasm to explain what all are there inside the cytoplasm, let me explain to you that one more thing about this biomembrane or cell membrane. So what happens, as you know that when your father or somebody builds a house, then there must be some window, there is a door. Why? Because there is a continuous need of something to come inside the room and go out of the room, because cells cannot survive on their own. They require certain things to survive. For example, they require glucose molecule. Why do cells require glucose molecule? To perform respiration reaction. And why do cells need to perform respiration reaction? To make ATP. And why do cells require ATP? In order to perform various kind of work. ATP is essentially the energy currency for the cell. Whenever ATP molecules break down into ADP and inorganic phosphate, because of gain of entropy, so what happens? Entropy increases, and when this entropy increases, there is a release of free energy. That free energy can be utilized by the cells to perform various function. So that's why cell would like a continuous supply of glucose. Second thing, cells would like a continuous supply of water. They require water. Second thing, cells require a continuous supply of amino acids. Why? Because they add up amino acids and then form protein. This is another matter that protein dissociate. They give amino acid, and the same amino acids can be used to form another protein, and then protein forms, do their function, and then protein has a lifetime. They again dissociate, and then the same amino acid. But there are some essential amino acids that are required by the cell to perform some essential function. Now, having said that, so cells also need a continuous supply of other ions like magnesium ions, like sodium ions, like chloride ions. And how would the cell get it? The cell would get it from outside only. Okay, if you talk about our body, then what happens? We eat certain things, and when we eat, let's say you have eaten some potato has starch, so 30% of starch will be digested in your mouth, and then rest of the starch and same thing again will go inside your stomach, and in the small intestine starch will be digested into glucose molecule or fructose molecules. When I say digestion essentially means breaking down the complex molecule, which is glucose, into smaller molecules, complex molecule like starch or some other polysaccharide into a smaller its component. Like digestion of carbohydrate would essentially mean that breakdown of starch into smaller monomers. Monomers are fructose and glucose and mannose, these things. If I talk about digestion of protein, then digestion of protein essentially means that protein molecules are broken down into amino acids. If I talk about fat, then fat are when you eat some oil or some oily thing, then oils are broken down into fatty acid and glycerol. Now the question is, why do we need digestion? Why? The first of all, everything that we have is a consequence of natural selection and evolution, so everything is useful, is needed. So just because we cannot absorb this complex molecule of carbohydrate or protein, that's why it has to be dissociated in our gut, in the intestine, so that it can take a form, a smaller form, which can be absorbed by the through the cells surrounding the intestine. So okay, I'm talking about plasma membrane, and I just told you that plasma membrane cells cannot survive on their own. They require certain things from outside. So how things from outside goes inside the cell? So I talked about this process. When we eat something, then we digest complex macromolecule like carbohydrate, like fat, like protein into smaller things like glucose, like mannose, like amino acids like fatty acid and glycerols, and these things gets absorbed through the cell into blood, and from blood blood circulation happens, and these things reach to each and every cell surrounding our body, and wherever it goes, so from blood these molecules of glucose and amino acids has to be absorbed inside the cell. Now you remember that glucose is a polar molecule. Now you remember amino acid is a polar molecule. Now these polar molecules, how would they cross a barrier of plasma membrane which has a two-layer of hydrophobic fatty acid, the lipid, two layer of lipids. So this thing is essentially fatty acid only. So and then you have it's like this, and this pinch and this pinch of mine is this fist of mine and this fist of mine are phosphate group, and these moving fingers are the lipid. So since this lipid is very much hydrophobic, your hydrophilic compounds cannot just cross through the layer of this phospholipid. So for passing of polar amino acids, polar water molecules, polar glucose molecules, there has to be some window and door on the plasma membrane. So nature designed in a way, nature designed in a way that they put lot of protein molecules at different places. Some protein molecules acted like a tunnel, and through the tunnel water molecules started coming inside the cell, depending on some other physics based on some concentration gradient that I'll talk later. And if sodium has to come inside, there has to be channel for sodium, there has to be gate for sodium ion, there has to be gate for potassium ion, there has to be a channel or gate for chloride ion, there has to be a chain gate for coming for this glucose molecule, there has to be a tunnel for amino acid transfer from outside to inside. And who make all this window and gate? So nature designed different kind of protein that are present on the plasma membrane for the transfer of required molecule from outside to inside, as well as from inside means cytoplasm, from cytoplasm to outside environment, as well as from outside environment to the cytoplasm. So this is the significance of plasma membrane. There are a lot to talk about it, that how much flexibility is needed, why plasma membrane is more flexible, how flexibility of plasma membrane keep on changing with time. So these are details, but on essence, now I explain to you what does a biomembrane essentially mean. Now I'll go, since I defined the boundary of the cell, now I will go into the cytoplasm and I'll talk about what all things are there inside the cytoplasm. Okay, so cytoplasm, main function of cytoplasm is to provide a controlled environment to perform different kind of reaction. Okay, so when I say controlled environment, I'll keep on giving 2-3 examples. Okay, so nature designed okay, so if I talk about your study room, would you like your study room is filled with some acid, or with some some base, or maybe would you like it to be meshed up all around? Although nature would like it to be meshed up, because nature favors higher entropy, nature favors disorderness. Having said that, essentially your cell environment, cytoplasmic environment, requires a controlled environment in terms of proton concentration. Proton concentration is essentially the pH of your cytoplasm. So nature has, this is a two-way thing. One statement, nature has designed cytoplasm to maintain a pH of 7.2 or 7.4 to perform different kind of metabolic activity. Another statement, since cytoplasmic pH was around 7.2, 7.4, so all the metabolic reactions evolved to perform their function and evolved to do such reactions in a pH of cytoplasm which is 7.2 or 7.4. So, but I would go with the second statement because it makes more sense. So things evolve as per the environment. So I am talking right now, I am talking in an English language just because I am in an English environment, and I can talk in Maithili because I have been in a Maithili environment, but I cannot talk Spanish, or I cannot talk in Japanese because I have not born and brought up there. So my language evolved I developed this communication skill in English or Maithili or Hindi because I have been in that particular environment. In a similar way, nature evolved everything in a particular environment, and probability works in a way that this environment suited best to perform these metabolic activities, and today we are alive. But yes, so there are constraint. What do I mean by constrained environment inside the cytoplasm? So it has some fixed pH, proton concentration. It has some fixed ionic concentration which decides its osmolarity. Like it has some fixed I guess 140 millimolar of potassium ion concentration, some, I'm not sure, but 150 millimolar of sodium ion concentration. It has some fixed concentration of chloride ion, calcium ion, and all. Second, and what if there is no boundary? All will be mixed into the in the water, and they'll not be able to perform activity away from, like this room. I am doing this experiment in a isolated room, but let's think that I am doing same thing in a market, sitting beside a wine shop. Would I be able to do this experiments efficiently if I'm sitting in an open environment near a wine shop or near a vegetable market? I won't be able to do this kind of experiment in that environment. So in order to perform certain work, you need a separate kind of environment. So, and that separate environment is the cytoplasm in terms of pH, in terms of ionic concentration, in terms of crowding, in terms of lot of other things. So during the evolution, when prokaryotic things formed at that time, so those prokaryotic organisms had one single, you know, single cytoplasmic environment where all the functions were getting performed. But then cells evolved into a form where they developed compartmentalization. So nature designed a different separate compartment to perform different kind of activity. So to make you understand this logic, let me tell you, what if I'm doing this experiment here? What if, I work in different labs. I do cell culture in cell culture lab. I do microinjection in microinjection room. I do laser lab experiments in FCS experiments in the laser lab. I do, if I have to do CD experiment or fluorescence experiment, I'll do in instrument lab. If I have to make some protein and purify it, I'll go in biochemistry lab. So there are different labs. Just imagine that everything is at a one place. For me, it would have been very easy, but there would be sharing, and there will be multiple clashes, and that would have not allowed me to talk to you in that way. All I am saying that so to perform different activity, nature designed different sort of environment, and it is same as making different room in your house. For cooking purpose you have different room, for reading purpose you have different room, and for bathing purpose you have different room, for sleeping you have different bedroom. So in the same way, in order to perform different reactions, cells have evolved different compartment. So in the eukaryotic, and it was not there in the primitive prokaryotic organism. It is there in the confined environment of your eukaryotic cytoplasmic environment. So if you have to perform some respiration reaction, then you have a different compartment, and that compartment is called mitochondria. So what is the function of mitochondria? Mitochondria takes some bigger molecule, and which is pyruvate. So pyruvate, what happens that when you eat your food, let's say you have eaten some potato, and then potato has starch is digested into glucose is absorbed into your blood, and from blood through it circulated in whole body, and in all of your cells on the plasma membrane there are protein molecules sitting on the plasma membrane, and these are called glucose transporter protein. So glucose transporter protein has some, so this protein is like this, and it is closed, and when there is a glucose it will just open like this. So if you can see, it can open. Okay, can I show? Okay, so I can show you like this. So this is your, let's say this, both of my hand here and here, both of my hand is your plasma membrane, and this is the protein, glucose transporter. So when glucose comes, it opens, and through this glucose molecule will come inside the cell. Inside the cell there are different enzymes, and these different enzymes perform different activity. And what happens, glucose is first of all, as soon as glucose enter inside the cell, first work is just to label it with some phosphate group, and they make it glucose at the 6th position of carbon. They add some phosphate group, and they make it glucose 6-phosphate. Why? So that glucose molecules become more polar when you add some phosphate group, and it does not it cannot escape out of the cell. So just if something comes in, just capture it, so that it does not go out. So using some kinase molecule, we call it hexokinase or glucokinase. So these glucokinase or hexokinase molecules add some phosphate group on the glucose and make it glucose 6-phosphate, so that it becomes more polar and charged, and it cannot escape out of the cell, because cells do not have protein transporter for the transfer of glucose 6-phosphate. But if it is glucose, they may escape out, and from then they do some Again further glucose 6 phosphate becomes fructose 6 phosphate, and then some enzymes come, they cut one covalent bond, breaks one covalent bond and forms two molecule, and at the end DHAP and some other molecules come, and at the end they form some pyruvate molecules, and these pyruvate molecules enter inside your mitochondria, and in the mitochondria oxidation happens. When I say oxidation, I essentially mean that loss of electron. So when loss of electron happens, then when you burn some organic molecule, let's say you are burning some wood, then what form would it take? If you burn a wood, burning essentially means combustion. Combustion essentially means oxidation. So oxidation of wood gives you some CO2 and water molecules. If you combustion of any organic molecule would give you or hydrocarbon, glucose is a hydrocarbon, would give you CO2 and H2O. In the same way, when combustion or oxidation of glucose molecule happens in the cytoplasm and further in the mitochondria, they would give you CO2 and H2O. But one thing remember, when you burn wood outside outside your body on the ground, then you get CO2 and H2O, and since it happens in one step only, in one step, oxygen oxidizes the carbohydrate or the hydrocarbon, and gives in one step, you form CO2 and H2O, and hence there is a huge gain of entropy, and whenever entropy increases, you release lot of energy, and so when you are burning a wood outside your body on the floor, then you are doing it in a one step, and hence all the energy is released in one step, and that we call heat. So lot of heat is gained when oxidation of hydrocarbon or carbohydrate happens in one step. But inside the cell, you do not want to oxidize your glucose that has come from your intestine to blood and blood to cell. You don't want to oxidize them in one step. If you would do that, then there is a huge loss of energy in the form of heat, and that will make you feverish, and you would not want to raise your temperature. Why? Because temperature is also a concern in that confined environment. Our protein molecules in our body, enzyme molecules in our body, works its best at 37 degrees centigrade, and if fever happens, your body temperature goes up, then performance activity of these protein molecules decreases, and you would not want that. So nature designed the oxidation of glucose molecule in multiple steps, so there will not be fully oxidation in one step, but rather there will be multiple loss of electron as per the gradient of electronegativity. Like oxygen is the whenever oxidation happens, then oxygen is the eventually ultimate acceptor of electron. Okay, oxygen accepts the electron. Why? Because oxygen has more electronegativity, and glucose molecule, when I talk about glucose molecule, so glucose molecule is C bonded hydrogen atom and then oxygen atoms. So carbon molecules carbon atoms has a low electronegativity. Hydrogen has low electronegativity. So whenever this electron from low electronegative atom goes to the higher electronegative atom, here oxygen, then electron is more stable being with a higher electronegative atom. So in oxidation, what happens? Oxygen is a more electronegative atom, and in glucose, carbon and hydrogen is low electronegative atom. So electron is more happy with oxygen, electron is less happy with carbon. When I say more happy means more stable means low energy. Okay, essentially when I say why, the question should be why is electron more stable with oxygen, which is a highly electronegative atom. The answer is because highly electronegative atom attracts electron more. When they attract electrons more, there is a more release of energy. Whenever bond forms, electrostatic, covalent, or any kind of bond forms, when attraction things happen, then energy releases, and release of energy makes it more stable. When an electron is with a more electronegative atom, they release more heat, and that more release of heat makes it more stable as compared to when it is with a lower electronegative atom like carbon and oxygen. So generally, when you, let's say, you burn glucose molecule outside in the environment, then you are essentially losing all the energy in one step, and it gives you heat. Okay, but inside the cell you would not want, you don't want to do that. So what happens, that on the membrane of mitochondria, there are multiple proteins, and these multiple proteins from here, so this thing is oxygen, this thing is glucose, and I'm putting it up considering that it is less stable. Less stable means more energy, and I am putting it here considering oxygen has when electron is with oxygen, then it is more stable, and when it is more stable, then essentially low in energy. So nature designed mitochondria in such a way that there are multiple protein molecules present on the membrane of mitochondria, such that it carries elements and metals in a gradient of electronegativity. So oxygen has the most electronegativity, carbon has the least electronegativity. Carbon will lose its electron to something else, this will lose electron to something else, this will lose electron to something else, and down the line, electronegativity of each protein which carries different kind of metals will keep on increasing, and hence electron is accepted by little more negative atom. It is accepted by little more electronegative atom, and hence energy which has to released when electron goes from highly electronegative low electronegative atom to highly electronegative atom, then energy releases. But inside the cell, nature designed it in a way that energy gets released into multiple steps, and when the energy gets released into multiple steps, this small energy is gained to form ATP molecule. So how does this happen? ADP molecule, ADP is adenosine diphosphate, they make covalent bond with inorganic phosphate and forms ATP. Now you remember here in this process, when ADP and inorganic phosphate associate with each other and ATP forms, then entropy reduces. And you must be knowing that reduction of entropy is not spontaneous. It requires certain energy. Now you must have understood the reason behind this oxidation of glucose, because when oxidation of glucose happens in multiple steps, energy releases, and this released energy is used by ADP and inorganic phosphate to form ATP molecule. ATP molecule of course has less entropy as compared to free ADP and inorganic phosphate molecules, and reduction in entropy for a reaction which show reduction in entropy, that must require energy from the outside. Now and how energy can be released from outside? Whenever there is a gain of entropy, energy will be released. So what is happening here? Here glucose molecule C6H12O6 molecule. Here glucose molecule C6H12O6 molecule is forming one molecule of glucose If you look at the whole respiration reaction, C6H12O6 plus 6H2O is giving you 6 molecule of CO2 and I don't know, 12 molecule of H2O. You balance it. Essentially, number of molecule in a respiration reaction increases. On the reactant side, you have only 7 molecules. On the product side, you have 12 molecules, or say 6H12O6 plus 6O2 gives you 6 CO2 plus 12 H2O. Yeah, whatever. So number of molecule increases in a respiration reaction, and because of this gain of entropy, because of gain of disorderness in respiration reaction, energy is released. Now I explained release of energy in two ways. One is when electron is going from low electronegativity to atom to high electronegative atom, then electron is more stable with high electronegative atom, and hence energy is released. And number two, whenever there is a gain of entropy, means gain of disorderness, energy will be released, and this released energy is accepted by adenosine ADP, adenosine diphosphate, and inorganic phosphate, and they forms ATP. In this reaction, ADP plus inorganic phosphate forming ATP, entropy is reduced, and when entropy is reduced, then it requires energy from outside, which they get from the this oxidation of glucose molecule. So in the same way, there is one more reaction. There is one more and one more thing I must tell you, and I have to focus on my experiment as well. So one more thing I must tell you, that when I told you that glucose molecule donate its, so essentially when glucose molecule breaks into pyruvate molecule in the cytoplasm, and pyruvate goes inside the mitochondria, and pyruvate also has carbon, hydrogen and oxygen, so electron is still in the carbon, and oxygen is the final electron acceptor, which has a high electronegative atom. And I told you there are multiple protein molecules present on the membrane of mitochondria. So nature designed every organelle inside the cell with a boundary to do different reactions in different kind of environment. So the wall of mitochondria is also made up of same phospholipid bilayer. So there is one layer of phospholipid, there is another layer of phospholipid. Okay, so this thing is phosphate, this thing is phosphate, and there is lipid. So mitochondrial membrane is also made up of same phospholipid, which plasma membrane is made up of. So and in the way plasma membrane has different protein molecules on its surface, in the same way mitochondria biomembrane also has various proteins to perform various kind of function. And I told you in the beginning that mitochondria was itself bacterial aerobic bacteria, which was symbiotically engulfed by another cells, and that's why it is present till now. But that is another story. But the essence of this I am talking about, that plasma membrane is around mitochondria as well. So if you don't want to call it, you call it mitochondrial membrane. And there are two membranes in mitochondria, outer membrane and inner membrane. So outer membrane is smooth, inner membrane is rough. Okay, outer membrane, inner membrane has some we call it F1, F0 particles projected everywhere. Outer membrane does not have this thing. Again, outer membrane has some lecithin. Okay, inner membrane has cardiolipin protein. One more thing, that outer membrane is more permeable for everything. Inner membrane is less permeable. It does not allow everything to go inside the. So the evidence that mitochondria was once a prokaryotic organism is that it has its own genetic material, circular double-stranded DNA. It has its own ribosome machinery as well, sub 70S ribosomes. So if you compare the inner mitochondrial environment with the bacterial environment, it is almost the same. and that's why we hypothesize that it has been engulfed inside the cell by some means. Okay. Now there is one more organelle of similar kind which has a 70s ribosome and some genetic material into it. So that we call chloroplast. So chloroplast is one of the molecule which to perform certain kind of action. And you must be knowing chloroplast is present in plants, and they do this respiration reaction. So what happens, friends, that this thing inside the, so what happens in photosynthesis? In photosynthesis reaction, 6 molecule of CO2, 12 molecule of H2O mix with each other and forms 1 molecule of glucose and 6 molecule of water. So what happens that 18 molecules, 6 CO2 and 12 H2O, 18 molecules is giving you 1 molecule of glucose and 6 molecule of water, means 7 molecule. So in a photosynthesis reaction, you have 18 molecules on reactant side and 7 molecules on the product side. Okay, so what do you see here? Here also see that entropy is reduced. And as I said, rule of thumb, that whenever a reaction show reduction in entropy, reduction in disorderness, then there has to be a gain of energy from outside. And from where photosynthesis reaction gets its energy? From the sunlight. Okay, and as I told you, whenever there is a loss, so from where sunlight comes? Of course from the sun. So sun gives you energy. Sun gives plant some energy to perform this photosynthesis reaction, which essentially a reaction in which entropy is getting lost. So, and sun is releasing energy, it means there must be gain of entropy in the sun, and that gain of entropy happens because of nuclear fusion inside this in the sun. So nuclear fusion happens in the sun, there is a gain of entropy. Because of that gain of entropy in the sun, there is a loss of energy, and that energy is utilized by plant in the chloroplast to perform the photosynthesis reaction, which is the loss of entropy. Okay. Now, so this is another organelle I talked about, which is chloroplast. Next organelle is something called Golgi body. So Golgi body is nicely membranous. Again, it has also chloroplast also has 2 layer of membranes, biomembranes, made up of phospholipid, and I explained that how phospholipids are associated with each other. Outside also phosphate, inside also phosphate, and in the middle you have lipids. Now, third thing is this Golgi body. Golgi body is required for trafficking of important molecules in and out of the cell, essentially outside the cell. So what happens? Let's say your pituitary cells form some growth hormone, and then growth hormone has to be released out of the cell. And it has to, let's say it has to do some processing and all. So it has to go through the Golgi complex. So Golgi complex was designed by nature for the trafficking purpose. If you have to, let's say many times you have to cells make some hormone, and that hormone has to go outside and release into the blood. Now who will help a cell to do this activity? It is the Golgi body. So Golgi body helps a cell to release its component outside, or maybe I don't know that if it helps to bring outside molecules in or not. You can call it yes, because when outside molecules come, one way that it comes through different protein molecules which are present on the membrane. Like if you have to transport glucose molecule from outside to inside, then there are glucose transporter protein present on the membrane. But let's say there are other ways, and these ways are called endocytosis. And what happens in endocytosis? Your membrane, your biomembrane forms smaller vesicles, and your molecule of interest which want to come in, they have to first go into the vesicle formed by the so this is the membrane, and then this membrane forms some vesicles and take the molecules in the vesicles, and vesicles come in and fuses with Golgi body, and from Golgi body it again goes wherever it is required. Yeah, so this is Golgi body is also one of the membranous organelle which is required for the trafficking of material outside the cell or inside the cell, and these materials are the materials which cannot go in and out using protein channel or protein transporter which are present on the membrane. Why? The reason is very simple, because nature must have thought that it is more energetically expensible to send all this hormone and other compounds through the protein transporter. So it's better that we use this thing. We use some membranous organelle that we call, here it is Golgi body. Another thing is endoplasmic reticulum. Endoplasmic reticulum is also similar to Golgi body, and it is of two kinds. One, if it does not have any ribosome, then it is called smooth endoplasmic reticulum, and if it has ribosome on it, then we call it rough endoplasmic reticulum. And you must be knowing that ribosome is a membrane-less organelle which has if it is inside the cell, it has two components, a smaller component and a bigger component. So this thing, eukaryotic ribosome is 80S ribosome and prokaryotic ribosome is 70S ribosome. And if I am not wrong, eukaryotic ribosome is made up of 60S bigger subunit, and there is a bigger subunit, there is a smaller subunit. So it's like this, okay, or maybe like this. So if I consider this as a bigger subunit, so 60S and one is 40S. And in case of prokaryotic, it has 70S, which is made up of 40S. So eukaryotic ribosome is 60 and 40, and prokaryotic is 50 and 30. Let's cross check it. So S is basically it defines the Swedberg unit. It is unitary system for ribosome, and it essentially describes the size of the ribosome. And what happens that ribosome is a protein machinery where protein synthesis happens. I'll talk about ribosome in a minute. First I should talk about nucleus, and then I'll talk about ribosome. But let me finish the organelles that are present in the cytoplasm. Another organelle, so since we are talked about Golgi body and endoplasmic reticulum, there is associated vesicle that is called lysosome. So lysosome has some acidic hydrolytic enzymes which is used to So hydrolytic enzymes, what will it do? It will just break the covalent bond when required, and that requirement is something that I'll not go in detail. But whenever cells are required, it just bursts, and sometimes it's a suicidal bag of cells. When cell has multiple problems, then cells has to go through apoptosis, and lysosome helps in that also. So and these things are Golgi, endoplasmic reticulum, and lysosomes are associated with each other, because you know that these all are membrane organelles, and which is involved in membrane trafficking. As I said, that if something has to come from outside to inside, and if that hormone or that something cannot come through the membrane, the protein present on the membrane, then it forms some vesicles. Like it uses some part of biomembrane, plasma membrane, forms some vesicles, and it comes in, then goes to associate with the Golgi body, and Golgi body further is released into the cytoplasm. So the vesicles, lysosome is also a kind of vesicles, but present in all kind of cells, present all the time, and it has some hydrolytic enzyme in it. Apart from this, we have smaller organelles as well, and that something like centriole and centrosomes. Centriole and centrosomes help in cell division, and today, in respect of time, I will not talk about cell division. I will talk about cell division some day later. Today I will stick to cells only. and I'll finish this video with a very major organelle, or maybe we shouldn't call it organelle, because it's the regulator, controller of the cell, and that thing we call nucleus. So nucleus is one of the most important part of the cell, and nucleus has genetic material into it. And when I say genetic material, for us, for eukaryotic cells, genetic materials are DNA. For some viruses, eukaryotic materials are RNA as well. We call them RNA viruses. Like COVID is an RNA virus. Correct me if I'm wrong. COVID is an is RNA virus. But if you talk about our cell, then our cell has this thing called DNA. DNA is present in the nucleus, and when DNA is in active state means when it is so DNA is a very long structure. We call them chromatin, and if the part of DNA which codes for messenger RNA or protein, we call them gene. And when gene is in active state, when I say gene is in active state, it essentially means that when gene is continuously making RNA, messenger RNA, and messenger RNA goes out and form protein. That is another thing. But part of if a part of DNA is active and doing its transcription, then DNA has to be in open state. But when at the time of cell division, these DNA molecules, which is acidic molecules, because it's essentially DNA is acid only, deoxyribonucleic acid. So this at the time of cell division, what happens that these DNA molecules wrapped around other positively charged protein molecules, we call them histone molecules, and also some non-histone proteins are there, which is required. Non-histone proteins are multiple other proteins, and those are not histones. Okay, so at the time of cell division, for the sake of equal distribution of genetic material among different cells, what happens that these cells, this DNA wrapped around some positively charged histone molecules, histone protein, and form chromosome like structure. Today I'll not talk about how does chromosome look like. I'll also not talk about what is chromatin, what is DNA, but I have to end this video with respect to time just by naming it like DNA genetic material is present in the nucleus. And what happens that this genetic material gene in the DNA forms the messenger RNA. Forms means it has the sequence of ATGC, adenine, thymine, guanine, cytosine, which sequence essentially defines the sequence of amino acid in the protein through messenger RNA. So inside the cell, in the nucleus, DNA, which has the sequence of ATGC, adenosine, thymine, guanine, cytosine, is transcribed into messenger RNA, and messenger RNA also has sequence of adenosine, no thymine but uracil, guanine, and cytosine. And this sequence of adenosine, uracil, guanosine, and cytosine, DNA has thymine and RNA has uracil. And if you compare the structure of thymine and uracil, you will find a difference of methyl group, and that makes RNA less stable and DNA more stable. And essentially, we do not want a stable RNA molecule, because function of RNA molecule is just to get the information from DNA and then get out of the nucleus, go to the ribosome, talk to the transfer RNA, and let them know the sequence of A, U, G, C. On messenger RNA, transfer RNA reads the sequence of A, U, G, C this sequence, and there can be multiple A, G, U, C, G, U, G, C, A, C, except T. There will be any sequence available on the messenger RNA. Transfer RNA reads this sequence, and according to the sequence of AUGC on the messenger RNA, they decide the sequence of amino acid in the protein. So, sequence of amino acid in the protein is decided by the sequence of AUGC on the messenger RNA, which in turns decided by the sequence of ATGC, which is present on the DNA. So this is this is called central dogma. So in central dogma, DNA to RNA and RNA to protein, that is happened. I want to talk about the difference between gene, different allele, chromosome, chromatin, and there is one more term chromatid. I'll talk about this in the next video. I also want to talk about how transfer RNA reads the sequence of amino acid on the messenger RNA and forms the sequence of protein accordingly at the ribosome. This also I'll talk in the later. Next, I want to talk about the structure of DNA, structure of messenger RNA, structure of ribosomal RNA, and structure of transfer RNA. I'll talk about it again in the next video. But one thing I did not talk, that is the nucleolus. So nucleolus is also a small structure, membrane-less structure inside the nucleus. It is form of some ribosomal RNA and some protein. It has some function that you have to read by yourself. And so this is the first video of cell, whatever you add, just an introduction of cell. I have to make another 2 videos on it. I'm thinking that first to define the structure of all these biomolecules, DNA, RNA, different kind of RNA, messenger RNA, transfer RNA, and ribosomal RNA, and third is the structure of protein and how transfer RNA decide how transfer RNA reads the sequence of AUGC on messenger RNA and decide the sequence of amino acid in the protein. Having said that, I started the theory, this video, by saying that 15 billion years ago there was a big bang. The whole universe was confined at a single point, and then this big bang happened. So you must have had a question that what was before big bang? Friends, there was no before, because time itself started with big bang. Thank you.